I always associate calcium with bones and teeth. So is it important in the function of the heart as well?
Yes, it is. Just as you said, most people think of calcium as helping build strong bones. In fact, calcium ions in solution perform all kinds of functions, and not only in the heart. They perform functions in cells throughout the body.
Next, let's look at some unique research in which scientists are using fluorescent proteins to light up calcium ions.
The Research Institute for Electronic Science at Hokkaido University. A research team here has been refining fluorescent proteins to illuminate in detail the function of calcium ions. It is led by Professor Takeharu Nagai.
Calcium ions perform an extremely important function in the exchange of signals between cells. Signal exchange occurs during all kinds of physiological functions. Our research aims to understand physiological phenomena by capturing minute fluctuations in calcium concentrations with a high degree of sensitivity.
Nagai-san's team developed a fluorescent protein called chameleon nano, the most sensitive ever devised. It reacts with even tiny amounts of calcium ions and instantly turns red. It was given the name chameleon nano because it changes color in response to the concentration of calcium ions. As the concentration rises, it changes from blue to yellow and then to red. Nagai-san's team has used chameleon nano to observe the concentration of calcium ions. That opens a window on signaling among cells. Their experiments used a remarkable organism, a cellular slime mold. Slime mold is normally scattered into separate, independently moving amoebas. But when nutrients become scarce, amoebas gather in one spot. 100,000 individual amoebas may merge into a single slug-like form that moves to seek a new habitat. What is the role of calcium ions when these primitive, single-celled organisms suddenly group together and act in unison? First, let's examine slime mold under natural light. The amoebas are already starting to assemble at a single spot. How does each individual know which way to go? Now, let's watch with chameleon nano lighting up the amoebas. You can see that the areas of higher calcium ion concentration spiral outward from the center. The individual slime mold amoebas gradually move towards the center of the spiral pattern. These are complex spiral patterns generated by signaling among individual slime mold amoebas. It's the first time this phenomenon has been observed.
Capturing footage of the signal propagation pattern was an impossible feat using traditional calcium tracers. This achievement perfectly illustrates that a picture is worth 1,000 words.
Another researcher is seeking medical applications for chameleon nano. Katsuhiko Mikoshiba. He is known worldwide for discovering the mechanism within nerve cells by which calcium ion concentrations change. It came about through his research on the brains of mice with motor ataxia. He compared the types of proteins found in the cerebellums of normal and impaired mice. The results showed that mice with motor ataxia are lacking just one protein, the IP3 receptor protein. The IP3 receptor protein is found on the surface of the endoplasmic reticulum inside the cell. Each of these black dots is an IP3 receptor. Their function is to regulate calcium ion concentration. Here is the mechanism that Mikoshiba-san discovered. A stimulus from outside the cell causes the cell membrane to produce a molecule called IP3. When IP3 binds to IP3 receptors, calcium ions are released from the endoplasmic reticulum. Without the IP3 receptor, cellular calcium ion concentrations are uncontrolled, so a mouse can't coordinate its movement. Using Chameleon Nano, Mikoshiba-san succeeded in capturing the change in calcium ion concentration. He believes that this method will be of value in researching neurological diseases.
There are many diseases, such as Alzheimer's, Huntington's, Parkinson's, and ALS, for which no treatment has been developed. My dream is that our work might greatly smooth the way for the development of treatments for these diseases.
The fluorescent protein Chameleon Nano could help researchers find cures for all kinds of neurological diseases. That's vital research.
Previous indicator compounds were only able to capture calcium ion concentration changes in cells such as cardiac muscle cells, where they occur on a large scale. But they could not accurately show small changes, for example, the changes that occur in nerve cells. Using Chameleon Nano, I believe we will be able to make what is happening in neurological disease clear.
You've been using bioimaging for a while now. Can you explain how you feel to be using this technology?
Well, for one thing, you can actually see the disease pathology literally unfolding with your own eyes. You can see the changes in the cells and blood vessels, so it is very different from looking at a graph or something. I have high hopes that these advantages will be exploited in the future, that they will help researchers understand and tackle diseases that continue to affect countless patients.
The original fluorescent protein used in bioimaging was discovered by a Japanese researcher, Osamu Shimomura. He received the Nobel Prize for that work in 2008. Japan seems to have a knack for this line of research.
Yes, I would have to agree with that. Japan also is very strong in optics technology, lens making, and other industries that support bioimaging. This is an industry that is well established in Japan, which means that Japanese researchers and scientists are able to rely on a very strong foundation.
Takamatsu-san, where do you hope to take your research from here?
Yes, well, what I am doing is building tools. I am building these tools to enable researchers to do new things. I want them to be able to see new things. Light comes in many, many forms that we haven't yet learned to harness. I want to create tools that see light in new ways to observe the activity of cells inside our bodies.
Fascinating. We wish you the best of luck. Today, we've been considering the medical applications of bioimaging. Yasu-san, what are your thoughts?
It sounds as though there are so many things we never knew before that we know now. And it's all thanks to this power to make cells glow so that we can actually see them working inside living tissue. I'm looking forward to hearing about further discoveries and more new research.
When you can actually see something with your own eyes, you can figure out what's really happening. There are tremendous implications for medicine, and Japan is very strong in bioimaging, so I have high hopes for what the future will bring.
Today, we focused on bioimaging. I'm sure we all sincerely hope that this field of research will yield better health for everyone in the future. That's all for Science Zero.
See you next time.